Welcome to the deep dive. Today we're getting right down to the chemical core of life itself.
We really are. We're looking at the molecules that define structure, function, heredity, everything.
We're talking about nucleic acids, the astonishing chemical basis of, well, you.
And maybe we should start with that powerful quote from our sources. It sort of sums it all up.
Yeah, go for it.
I am the chemical basis of life and heredity, organized into genes that control every aspect of cellular function.
Powerful stuff. And that I is, of course, DNA, deoxyribonucleic acid.
The one and only.
So our mission for this deep dive is pretty clear. We want to give you a really solid grasp of these fundamental molecules, DNA, RNA, and their building blocks, the nucleotides.
Right. Think of this as your shortcut to understanding the central dogma of biology. We're connecting the tiny molecular pieces to the big picture of life.
It's funny, though, how recent all this knowledge really is in the grand scheme of things.
Absolutely. DNA itself was first spotted way back in 1869 by Johann Friedrich Miescher, but he just called it nuclein, something weird he found in cell nuclei.
So they knew of it, but not really what it did for ages.
For decades. It wasn't until, 1944? Avery, McLeod, and McCarty did those groundbreaking experiments.
The ones that showed DNA was the genetic carrier, not protein, which was the prevailing theory back then.
Exactly. That experiment really kicked off the whole molecular revolution, paved the way for the 50s and Watson and Crick, #TechI, the fundamental players in the central dogma.
Okay, so let's lay out the main players. We've got two types of nucleic acids.
DNA, deoxyribonucleic acid, and RNA, ribonucleic acid.
And their core job is, well, it's information, right? Storing it and transmitting it.
Precisely. DNA is like the master blueprint, or maybe the reserve bank, as you put it earlier. It's for long-term stable storage.
Holding the hereditary information, keeping species consistent over generations.
And containing the instructions that control, literally every aspect of how a cell works. Growth, energy, everything.
So DNA is the archive.
Yeah.
And the genes, they're the individual files within that archive, the basic units of information.
Right, which leads us directly to the flow of that information, the central dogma.
Ah, yes, the big one. RNA makes protein.
That's the core sequence. DNA, RNA, protein. DNA stays protected in the nucleus, mostly.
And it sends out RNA messengers with instructions.
Exactly. RNA carries the specific instructions to the cellular machinery that actually builds the proteins. Proteins do the work. That's the chain of command.
Okay, but before we get to the famous double helix structure, we need to understand what it's built from. The monomers.
The nucleotides.
Right.
Nucleic acids are polymers. Polynucleotides, long chains of these nucleotide units.
So what's in a nucleotide? Let's break it down.
It's got 3 essential parts chemically linked together. You've got a nitrogenous base, a 5-carbon sugar, that's the pentose sugar, and at least 1 phosphate group.
And there's a key distinction here, something that often trips people up. Nucleoside versus nucleotide.
Good point. A nucleoside is just the base plus the sugar. That's it.
Add the phosphate group, then it becomes a nucleotide.
Correct. Base plus sugar equals nucleoside. Base plus sugar plus phosphate equals nucleotide. And that phosphate group is really important, as we'll see. It gives the whole molecule its charge.
What's really cool, though, is that these nucleotides aren't just, bricks waiting to be stacked. They have jobs on their own.
Oh, absolutely. They're incredibly versatile. Their function goes way beyond just being building blocks for DNA and RNA.
Like ATP. Everyone knows ATP.
Exactly. Adenosine triphosphate. It's a ribonucleotide. And it's the universal energy currency of the cell.
Why? What makes it good for energy?
It's those phosphate groups, specifically the bonds holding the 2nd and 3rd phosphates on. They're high energy bonds, sort of unstable.
So easy to break.
Right.
Yeah.
When the cell needs energy, it just snaps off that last phosphate and boom, a burst of usable energy is released. It's immediate.
Okay, so the triphosphate structure is key for energy delivery. What else do they do?
Well, they're also crucial parts of coenzymes. Think about the B vitamins.
Right, like FAD and NAD plus tau.
Precisely. Flavin adenine dinucleotide, nicotinamide adenine dinucleotide. These are nucleotide derivatives. They're absolutely essential for metabolic reactions, particularly redox reactions carrying electrons around.
So they help enzymes do their jobs.
They do. The nicotinamide part of NAD plus scoring, for instance, is perfectly set up to accept or donate hydrogen, which is fundamental for pathways like respiration.
And you mentioned signaling, too.
Yes. They act as intracellular messengers. Cyclic AMP and cyclic GMP, cGMP are classic examples.
How does that work?
Well, imagine a hormone arrives at the cell surface. It can't get in, so it triggers the production of CAMP inside the cell. CMP then acts as a second messenger, spreading the signal internally and activating a whole cascade of responses.
Wow. So without nucleotides, the cell would basically fall apart. No energy, trouble with metabolism, no internal communication.
Pretty much. They are fundamental in so many ways beyond just genetics.
Okay, let's dig into those three components more deeply, starting with the nitrogenous bases. They're ring structures, right?
Yes, aromatic heterocyclic compounds, meaning they have rings containing atoms other than just carbon, specifically nitrogen, and they have that stable aromatic electron structure. They fall into two main classes based on their structure.
Purines and pyrimidines.
Correct. The carins are the bigger ones with a double ring structure that's adenine A and guanine G. You find both of these in DNA and RNA.
And the pyrimidines, smaller single rings.
Right. Cytosine C is one, and it's also found in both DNA and RNA.
Okay, so A, G, and C are common ground, but here comes a key difference between DNA and RNA.
This is a big one. It involves the second pyrimidine base. In DNA, it's thymine T.
But in RNA, it's uracil U.
Exactly. T is in DNA, U in RNA. Structurally, they're very similar. Thymine just has an extra methyl group, a CH3 group attached at the C5 position of the ring, which uracil lacks.
Seems like a tiny difference. Does it matter much?
Oh, it matters hugely. That little methyl group on thymine adds stability to DNA. It also provides a way for the cell's repair machinery to recognize and fix a common type of DNA damage where cytosine accidentally gets converted into uracil. If uracil were normally in DNA, it would be much harder to spot those errors.
Ah, clever. Okay, you also mentioned something called tautomerism earlier. Sounds complex, but you said it's important for stability.
It is. It's a concept from chemistry. Tautomerism means a molecule can exist in slightly different forms because a proton, just a hydrogen ion, can hop around.
So the atoms are the same, just arranged slightly differently.
Kind of. For these bases, it involves shifting a proton and rearranging some double bonds. This leads to two main forms: the keto form, which chemists also call the lactam form, and the enol form, or lactam form.
Which bases do this?
Cytosine, guanine, thymine, and uracil all show this keto-enol tautomerism. Adenine does too, but it's called amino-imino tautomerism. The principle's the same.
So why is this important for DNA? Why can't they just flip-flop between forms?
Because the whole Watson-Crick base pairing, the AT and GC pairing that holds the double helix together, relies on the bases being in a specific tautomeric form. If they were constantly flipping, they wouldn't pair up correctly. I might try to pair with C sometimes, for example.
Which would mess up the genetic code. Mutations galore.
Exactly. Chaos. But biology has it sorted. At the normal pH inside a cell, around 7.4, the chemical equilibrium strongly favors one form, the keto or lactam form for C, G, T, and U, and the amino form for A.
So they mostly stay locked in the form that allows the correct hydrogen bonding.
Precisely. It ensures that A reliably pairs with T and G reliably pairs with C. It's a subtle chemical preference that underpins genetic fidelity. Amazing, really.
Wow. Okay, component 2, the sugar, another key difference here.
Yes, the pentose sugar. In RNA, it's D-ribose, a standard 5-carbon sugar.
But in DNA, it's D2-deoxyribose. What's the deoxy mean?
It means missing an oxygen. The difference is at the number 2 carbon atom of the sugar ring. We call it the 2' or C2 position.
Prime. Why prime?
Just a naming convention. To distinguish the carbons in the sugar ring, 1, 2, 3 for DART, from the atoms in the base ring, which just use numbers 1, 2, 3, etc.
Got it. So at C2, what's the difference?
Ribose has a hydroxyl group, an acyl group attached to C2. Deoxyribose just has a hydrogen atom, extra each. It's missing that oxygen atom.
And again, does that small change matter?
Absolutely. That missing oxygen makes DNA much less chemically reactive than RNA, much more stable over the long term, perfect for its job as the permanent genetic archive. RNA, being more reactive, is better suited for its shorter-term roles.
Makes sense. Okay, let's quickly touch on nomenclature, naming these things.
Right. Base plus ribose equals ribonucleoside. Like adenine plus ribose equals adenosine. Guanine plus ribose guanosine, and so on.
And base plus deoxyribose equals deoxyribonucleoside.
Exactly. Adenine plus deoxyribose equals deoxyadenosine. And we use the prefix sater for deoxy versions when we talk about the nucleotides.
So add a phosphate to deoxyadenosine and you get deoxyadenosine monophosphate, or DAMP.
Yeah.
Add one to adenosine, you get adenosine monophosphate, AMP. Okay. How do all three pieces, base, sugar, phosphate, actually connect? What kind of bonds?
They're covalent bonds, strong links. First, the base attaches to the C1 position of the sugar. This is called an N-glycosidic bond.
N-glycosidic.
Yes, because it forms between the C1 of the sugar and a nitrogen atom in the base ring specifically, the N9 nitrogen for purines or the N1 nitrogen for pyrimidines.
Okay. Base to C1 of sugar. Where does the phosphate go?
The phosphate group typically attaches to the C5 position of the sugar, forming an ester bond with the 5-hydroxyl group. So we usually talk about nucleotides being 5' monophosphates like AMP or DMP.
And if you add more phosphates, like for ATP?
They just get added in a chain onto that first phosphate at the 5' position. So AMP becomes ADP diphosphate, then ATP triphosphate.
And you mentioned the charge earlier.
Right. Each of those phosphate groups carries a negative charge at physiological pH. So nucleotides, and especially the long chains of nucleic acids like DNA and RNA, are highly negatively charged molecules. They're anions.
Which explains why they like to interact with positively charged things.
Like histone proteins, which we'll get to later when we talk about DNA packing. That charge interaction is absolutely key.
All right, we've got the building blocks, the deoxyribonucleotides, DMP, DGMP, DCMP, and DTMP. Now, how do they link up to form the long DNA polymer?
They join together via what are called 3', 5 types of phosphodiester bridges. It's a very strong directional linkage.
Okay, break that down. Phosphodiester.
Right. The phosphate group attached to the 5' carbon of one sugar forms an ester bond with the hydroxyl group on the 3' carbon of the next sugar in the chain.
So the phosphate acts as a bridge connecting the 5' carbon of one sugar to the 3' carbon of the next.
Exactly. And because it forms two ester bonds, one to the 5' sugar, one to the 3' sugar, it's a diester bond involving phosphorus, hence phosphodester. This creates the sugar phosphate backbone of the DNA strand.
And it's directional, you said. 3' and 5'.
Yes. Each DNA strand has an inherent directionality. One end has a free phosphate group attached to the 5' carbon, that's the 5'. The other end has a free hydroxyl group on the 3' carbon, that's the 3' end. We always read DNA sequences from 5 to 3'.
Okay. Now, before Watson and Crick figured out the 3D shape, there was a crucial piece of the puzzle from Erwin Chargaff.
Ah, Chargaff's rules. Yes, absolutely critical. In the late 1940s, Chargaff painstakingly analyzed the chemical composition of DNA from lots of different organisms.
And he found a pattern, right?
A consistent striking pattern. He found that in any sample of double-stranded DNA, the amount of adenine, A, was always almost exactly equal to the amount of cytosine,
T.
and the amount of guanine, G, was always almost exactly equal to the amount of cytosine, C,
Yes.
which also means the total amount of purines, A plus G, equals the total amount of pyrimidines, T plus C.
This couldn't be a coincidence. It strongly suggested A pairs with T and G pairs with C somehow.
It was a massive clue, the foundation for the base pairing model. But it's important to note these rules only apply to double-stranded DNA.
Right. Single-stranded DNA or most RNA molecules don't follow these rules because they don't have that second complementary strand.
Although, interestingly, some viruses have double-stranded RNA genomes, and those do obey Chargaff's rules. A pairs with U, G pairs with C.
Fascinating. Okay, so Chargaff sets the stage. Then come Watson and Crick in 1953 using X-ray diffraction data, especially from Rosalind Franklin.
And they proposed the double helix model for bDNA, the structure that won them the Nobel Prize in '62. It's become iconic.
The twisted ladder. Let's talk structure. Key features.
Okay, first and foremost, it's two strands, two polydeoxyribonucleotide chains coiled around each other.
And they run in opposite directions.
Yes, they are anti-parallel. One strand runs 5 to 3 foot, its partner runs 3 to 5 foot, like traffic on a two-way street.
Why is that anti-parallel arrangement so important?
It's essential for allowing the bases to pair up correctly in the middle and for the enzymes involved in DNA replication and repair to work properly.
Okay, what about the dimensions? Is it a regular structure?
Remarkably regular. The diameter across the helix is a constant 2.0 nanometers, or 20 angstroms.
And the twist?
Each full turn of the helix, the pitch, takes 3.4 nanometers, 34 angstroms. And within that one turn, there are exactly 10 base pairs stacked on top of each other.
So the distance between adjacent base pairs is 0.34 nanometers.
Precisely. Very tightly packed.
And where are the different chemical parts located?
Right.
The backbone versus the bases.
The sugar phosphate backbones, which are hydrophilic, water-loving because of all those charged phosphates, are on the outside of the helix, facing the watery environment of the cell.
And the bases.
The nitrogenous bases, which are relatively hydrophobic, water-repelling, are tucked inside, stacked flat like coins in the core of the helix, perpendicular to the axis. This shields them from water and stabilizes the structure through base stacking interactions.
Okay, now the crucial part. How are the two strands held together?
By hydrogen bonds between the complementary bases, this is where Chargaff's rules become physical reality.
A pairs with T, G pairs with C. How many
Yeah.
bonds?
Adenine always pairs with thymine, forming 2 hydrogen bonds. Guanine always pairs with cytosine, forming 3 hydrogen bonds. 3 bonds for GC, only 2 for AT. Does that make a difference?
Yes, a significant one. The text-hard pair is roughly 50% stronger, more stable than the texter pair. DNA regions rich in GC require more energy, like higher temperatures, to separate the strands.
And why those specific pairs? Why not A with C or G with T?
It comes down to two things: size and hydrogen bonding geometry. Remember, purines, Ag, are bigger than pyrimidines,
CT.
To keep that constant 2.0 millimeter diameter, you must pair a purine with a pyrimidine.
Ah, so 2 purines will be too wide, 2 pyrimidines too narrow.
Exactly. A purine-pyrimidine pair fits perfectly. And only the AT and GC pairings allow the hydrogen bond donor and acceptor atoms on the bases to align correctly to form those stable bonds across the helix.
So the complementarity isn't just adjusted by Chargaff's numbers, it's enforced by the physical and chemical constraints of the helix structure. Perfectly put, the strands are complementary, not identical. If you know the sequence of one strand, you automatically know the sequence of the other.
And functionally, one strand carries the actual code, right?
Usually, yes. We call that the template strand or sometimes the sense strand. The information on the strand is transcribed into RNA. The opposite strand is the antisense strand.
What about the grooves we see on models of DNA? The major and minor grooves?
Those are important too. They arise because the glycosidic bonds attaching the bases to the sugars aren't directly opposite each other. This creates a wider gap, major groove, and a narrower gap, minor groove, on the surface of the helix.
And proteins can access the bases there.
Yes, especially in the major groove, which is wider and exposes more of the edges of the base pairs. Many proteins that regulate gene expression bind to specific DNA sequences by recognizing the pattern of hydrogen bond donors and acceptors presented in the major groove, often without needing to unwind the helix first. #Tag5 meet DNA conformations, organization, and dynamics.
Okay, so bDNA, the right-handed double helix, is the standard model. But you mentioned DNA isn't always stuck in that one shape.
No, it's quite dynamic.
Yeah.
While bDNA is the most common form found in cells under physiological conditions, DNA can adopt other conformations. The main ones studied are aDNA and zDNA.
How does aDNA differ from bDNA?
aDNA is also a right-handed helix, but it's shorter and wider than bDNA. It has 11 base pairs per turn instead of 10, and the bases are tilted quite significantly relative to the helix axis, about 20 degrees.
When would DNA adopt the A form?
It tends to form under dehydrating conditions, so maybe not as common in the cell's watery environment. However, importantly, RNA-DNA hybrid helices, which form during transcription, adopt an A-like conformation. Double-stranded RNA also takes an A-form structure.
Okay, now zDNA sounds intriguing. The rebel helix?
That's the one. zDNA is radically different. First off, it's a left-handed helix, twisting the opposite way to A and B DNA.
Left-handed, wow.
And it's thinner and more elongated, with 12 base pairs per turn. The most striking thing visually is that the sugar phosphate backbone follows a zigzag path, which is where the Z's name comes from.
What makes DNA switch to this weird Z form?
It's favored by certain DNA sequences, particularly stretches of alternating purines and pyrimidines, like repeating CG sequences, poly-DGGC. Also, things like high salt concentration or negative supercoiling can induce the beta-Z transition.
Does this structural change do anything? Is it just a curiosity?
Oh, definitely not just a curiosity anymore. It's thought that these transitions, especially beta-Z, might act as regulatory switches in the genome.
How so?
Well, zDNA has a very different shape, which could be specifically recognized by certain proteins. It might pop up transiently in regions under torsional stress during transcription, maybe helping to relieve that stress or signaling something to the cellular machinery. It's strongly implicated now in regulating gene expression.
Beyond these main helical forms, are there even more unusual structures?
Yes, particularly structures that are important for molecular recognition or that arise from DNA damage.
Like bent DNA.
Right. While the helix is often shown as a straight rod, it can definitely bend. Certain sequences, like tracks of consecutive adenines, A tracks, can cause slight bends. More dramatically, damage or binding by certain drugs can cause sharp kinks.
You mentioned cisplatin earlier, the anti-cancer drug.
Yes, cisplatin binds to DNA, often linking adjacent guanines, and causes the DNA to bend significantly into the minor groove. This distorted structure is then recognized by cellular proteins, which can trigger repair pathways or, ideally in cancer treatment, lead to cell death.
What about structures with more than two strands? Triple or quadruple helices?
They exist, using different types of hydrogen bonds, not just the standard Watson-Crick ones. These are called Hoogsteen base pairs, named after Karst Hoogsteen, who first described them. They allow a third strand to wind around the major groove of a standard double helix, forming triple-stranded DNA, also called hDNA. You might see triplets, like T pairing with an AT pair, or a protonated C pairing with a GC pair.
Are these stable?
Generally less stable than double helices, partly because you're bringing three negatively charged backbones close together, which causes electrostatic repulsion, but they do form under certain conditions, often in sequences with long stretches of purines on one strand and pyrimidines on the other. They might play roles in gene regulation or recombination.
And four strands, G-tetraplexes.
Yes, these are fascinating and clinically very relevant. They form in DNA regions that are very rich in guanine.
How do they work?
Four guanine bases arrange themselves in a square, hydrogen bonding with each other through Hoogsteen bonds. This square is called a G-quartet. Then, these G-quartets stack on top of each other, often stabilized by a positive ion like potassium sitting in the channel down the middle. This creates a stable four-stranded structure, the G-tetraplex, or G-quadruplex.
Where do we find these in our cells?
Most famously, they're found in telomeres. Those repetitive DNA sequences at the very ends of our chromosomes that protect them from degradation.
And why is that clinically relevant?
Because telomeres are maintained by an enzyme called telomerase, which is overactive in many cancer cells, allowing them to divide indefinitely. G-tetraplexes forming in telomeres can actually inhibit telomerase activity. So molecules that stabilize these G-tetraplex structures are being actively developed as potential anti-cancer drugs.
Wow. They're also implicated in HIV, you said.
Yes, G-rich sequences in the HIV RNA genome can form G-tetraplexes that seem to be important for the virus's life cycle, specifically in packaging its genome. So they're a target for anti-HIV therapies too.
Okay, to really appreciate all this structure and function, we need a sense of scale. How much DNA are we talking about?
The numbers get pretty big. We measure DNA length in base pairs, BP. 1,000 is a kilobase, KB. 1,000,000 is a megabase, MB. And 1,000,000,000 is a gigabase, GB.
Let's take an example. A simple bacterium like E. coli.
E. coli has a circular chromosome with about 4.6 million base pairs, 4.6 millimeters. If you were to stretch that circle out, its contour length would be about 1.5 millimeters. Manageable.
Now us, a human cell, diploid, so 2 copies of the genome.
We have about 3 billion base pairs per haploid set. So a diploid cell has roughly 6.0 billion base pairs, 6.0 gibbers.
And the contour length of that, stretched end to end.
An absolutely staggering 2 meters. 2 meters of DNA packed inside almost every single one of your cells.
2 meters. And that has to fit inside the nucleus, which is only about 10 micrometers in diameter.
That's right. A micrometer is a millionth of a meter. So you're compacting 2 meters of thread into a space about 10 millionths of a meter across. It requires an incredible packing ratio, something like 1,000,000 to 1.
How on earth is that achieved? It can't just be randomly crumpled up.
No, it's a highly organized hierarchical process. The key players in eukaryotes are positively charged proteins called histones.
Because DNA is negatively charged, opposites attract.
Exactly. The negatively charged DNA backbone wraps tightly around these basic, positively charged histone proteins. The combination of DNA and associated proteins is called chromatin.
And the fundamental unit of this packing is the nucleosome.
Yes, the nucleosome is the first level of organization. Think of it as a spool. It consists of about 147 base pairs of DNA wrapped almost twice, about 1.7 turns, around a core complex of 8 histone proteins.
Which histones are in the core?
2 molecules each of 4 types, H2A, H2B, H3, and H4. This octamer core forms the protein spool.
So the DNA wraps around this core. What does that look like?
Under an electron microscope, this first level of packing looks like beads on a string. The beads are the nucleosome cores, and the string is the short stretch of linker DNA connecting one nucleosome to the next. This structure is about 10 nanometers in diameter.
Okay, that's the 10 millimeter fiber. But that's still not nearly compact enough to fit 2 meters in a nucleus.
Right. So the next level involves coiling up that beads on a string fiber. A fifth type of histone, H1, binds to the linker DNA and helps the nucleosomes coil together into a thicker fiber, about 30 nanometers in diameter.
The 30-nanometer fiber, often called a solenoid structure.
Yes, a solenoid or sometimes a zigzag structure. It typically involves about 6 nucleosomes per turn of the coil. This represents a further significant compaction.
Even that gets folded further.
Yes. The 30-nanometer fiber is then organized into large loops, which are anchored to a protein scaffold within the nucleus. And these loops get further coiled and condensed, especially when the cell is preparing to divide, ultimately forming the highly condensed, visible chromosomes we see during mitosis. It's an amazing hierarchy of folding.
Just incredible. Okay, let's shift gears slightly to dynamics. The helix isn't static. It needs to open up sometimes. Denaturation.
Right, denaturation or melting. This is the process where the two strands of the double helix separate. It's usually caused by heat or changes in pH, extreme acidity, or alkalinity.
What actually breaks?
Importantly, it's the relatively weak hydrogen bonds between the base pairs that break. The strong covalent phosphodiester bonds along the backbone remain intact. So the strands separate, but they don't break into smaller pieces.
Can we observe this happening?
Yes, very easily in the lab using UV light. DNA bases absorb UV light strongly, particularly at a wavelength of 260 nanometers.
But there's a difference between double-stranded and single-stranded DNA's absorbance.
Yes, this is called the hyperchromic effect. When the bases are stacked neatly in the double helix, they sort of shield each other electronically, and their ability to absorb UV light is reduced. This is called hypochromism.
But when the helix melts,
And the strands separate, the bases become unstacked and are more exposed. As a result, their absorbance of 260 nanometer light increases significantly by up to 40%. This increase in absorbance hyperchromicity is a direct measure of how much the DNA has denatured.
and this relates back to the GC versus AT content.
Absolutely. Because GC pairs have 3 hydrogen bonds and are more stable than AT pairs with 2 H bonds, DNA that has a higher percentage of GC base pairs requires more energy, meaning a higher temperature, to melt.
So we can define a melting temperature, T.
Exactly. The Texi-pair is the temperature at which 50% of the DNA duplex has denatured into single strands. It's a characteristic property of any given DNA molecule and is directly proportional to its GC content. Higher GC content means a higher Texium. This is fundamental for techniques like PCR.
Okay, we've spent a lot of time on DNA, the master archive. Let's switch focus now to RNA, the versatile worker molecule. What are the key differences again? Let's recap.
There are 5 main structural differences to keep in mind. First, the sugar.
RNA has ribose, DNA has deoxyribose, that 2-bit hydroxyl group.
Right. Second, the bases.
RNA uses uracil U instead of thymine T. Pairs with U in RNA context.
Correct. Third, the overall structure.
DNA is typically a double helix. RNA is usually single-stranded.
Usually, yes, though RNA can, and often does, fold back on itself to form complex secondary and tertiary structures, including short double helical regions where complementary bases pair up, AU and GC, but it doesn't typically form long, stable double helices like DNA.
Okay, fourth difference.
Because it's mostly single-stranded, RNA generally doesn't obey Chargaff's rules. The amount of A doesn't necessarily equal U, nor G equal C.
And the fifth difference comes back to that 2-monohydroxyl group again. Stability.
Yes. That AOH group on the C2 monoribose makes RNA chemically less stable than DNA. Specifically, it makes RNA susceptible to hydrolysis being broken down under alkaline basic conditions. DNA, lacking that group, is resistant to alkali hydrolysis.
So RNA is inherently more transient, which fits its role as a messenger or temporary molecule.
Precisely. Its instability is actually part of its function in many cases.
All right, let's talk about the main types of RNA in the cell. There are 3 major players in protein synthesis. First, messenger RNA.
Messenger RNA, or mRNA. This is the transcript. Its job is to carry the genetic code for a specific protein from the DNA in the nucleus out to the ribosomes in the cytoplasm, where the protein will actually be made.
So it's the messenger carrying the instructions for one specific job.
Exactly. Each mRNA molecule typically codes for one protein in eukaryotes, or sometimes several related proteins in prokaryotes. In eukaryotes, it's initially made as a longer precursor molecule called heterogeneous nuclear RNA, or hnRNA.
And this hnRNA needs processing.
Yes, extensive processing. It undergoes splicing to remove non-coding regions, introns, and it gets crucial modifications at both ends for stability and recognition.
These are the cap and tail.
Right. At the 5' end, a special structure called the 5' cap is added. It's a chemically modified guanosine nucleotide, 7-methylguanosine, linked in an unusual 5 to 5 foot triphosphate bridge.
What does the cap do?
Two main things. It protects the mRNA from being degraded by exonucleases, enzymes that chew up nucleic acids from the ends, and it's essential for the ribosome to recognize the mRNA and initiate translation, protein synthesis.
And at the other end, the 3' end?
At the 3' end, most eukaryotic mRNAs get a poly-A tail added. This is a long string, typically 20 to 250, of just adenine nucleotides.
Poly-A. Just A, with its function.
It also contributes to mRNA stability, protecting the 3' end from degradation. The length of the poly-A tail can also influence how long the mRNA lasts in the cytoplasm and how efficiently it's translated.
Okay, so mRNA carries the code. What delivers the building blocks, the amino acids?
That's the job of transfer RNA, or tRNA.
The adapter molecule.
Exactly. tRNA molecules act as adapters that can read the genetic code on the mRNA and bring the corresponding amino acid to the ribosome. There's at least one specific type of tRNA for each of the 20 common amino acids.
What do they look like? Are they big?
No, they're relatively small, usually only 71 to 80 nucleotides long, but they fold into a very specific and conserved three-dimensional shape. If you draw it flat, it often looks like a clover leaf.
The clover leaf. What are the leaves?
The clover leaf structure highlights 4 main arms, which are short stems of double helix formed by base pairing, ending in loops of single-stranded RNA.
Which arms are most important?
Well, they all are, but 2 are key for the adapter function. At the top of the clover leaf is the acceptor arm. This ends with the specific 3-nucleotide sequence, CCA. The amino acid gets covalently attached to the adenine at the very end of the CCA sequence.
So that's where it carries its cargo. What's the other key arm?
Opposite the acceptor arm is the anticodon arm. This contains a loop with 3 specific bases called the anticodon.
And the anticodon reads the mRNA.
Yes. The 3 bases of the anticodon are complementary to a 3-base codon on the mRNA molecule. This codon-anticodon pairing is what ensures the correct amino acid is brought in according to the mRNA's instructions.
What about the other arms? They have weird names in the outline. D arm and TP-CC arm.
Right. Those arms are named after unusual modified bases that are often found in their loops. The D arm frequently contains dihydroidan D, and the TC-EAT arm contains ribothymidine T and pseudoridine. That's the Greek letter PSI.
Modified bases, so not just AUGC.
Correct. tRNAs are famous for having a variety of chemically modified bases created after the tRNA is initially transcribed. These modified bases are crucial for giving the tRNA its correct, stable 3D structure, which is actually more like an L shape than a flat clover leaf. That specific shape is needed for it to fit into the ribosome properly.
Okay, mRNA brings the message, tRNA brings the amino acids. Where does the actual synthesis happen?
On the ribosomes. And ribosomes themselves are made largely of the 3rd main type of RNA, ribosomal RNA, or rRNA.
So RNA is structural.
Yes, primarily. rRNA molecules, together with numerous ribosomal proteins, form the 2 subunits of the ribosome, the large subunit and the small subunit. RNA actually makes up the bulk of the ribosome's mass, often 60% or more.
What are the RNA types in eukaryotes?
Eukaryotic ribosomes are designated 80S. S is a unit of sedimentation. They consist of a large 60S subunit and a small 40S subunit. The 60S subunit contains 3 RNA molecules. 28S, 5.8S, and 5S are RNA. The 40S subunit contains 1, 18S RNA.
So several different RNA molecules work together. Do they just provide scaffolding?
For a long time, that's mostly what we thought, but turns out RNA does much more than just provide structure. It plays active roles in binding the mRNA, binding the tRNAs, and this was a huge discovery, actually catalyzing the formation of the peptide bond that links the amino acids together.
Wait, the RNA itself catalyzes the reaction, like an enzyme.
Exactly. This brings us to the concept of ribozymes.
RNA enzymes.
Yes, it turns out some RNA molecules can act as biological catalysts, just like protein enzymes. The peptidyl transferase activity, the formation of the peptide bond within the ribosome, is catalyzed by the 28S RNA in the large subunit, not by any of the ribosomal proteins.
That's incredible. Are there other examples of ribozymes?
Oh, yes. Another classic one is RNase P, an enzyme involved in processing tRNA precursors. It has both protein and RNA components, but the RNA component alone is catalytically active. There are also self-splicing intron segments of RNA that can catalyze their own removal from an mRNA precursor.
This discovery must have had huge implications for thinking about the origin of life.
Immense. It led to the RNA world hypothesis. The idea is that early in evolution, before DNA and complex proteins existed, RNA might have served as both the genetic material and the main catalytic molecule.
So RNA could store information and perform enzymatic work.
Potentially, yes. Ribozymes show that RNA can be catalytic. It suggests a plausible scenario where life started with simpler RNA-based systems before evolving the more stable DNA for information storage and the more versatile proteins for catalysis.
Mind-blowing. And you mentioned this catalytic ability has modern medical potential.
It does. If RNA can be designed to cut other RNA molecules, maybe we can design specific ribozymes, sometimes called aribozymes, when they're engineered to target and destroy faulty mRNA molecules that cause disease.
So if a cancer cell is overproducing a certain mRNA, you could introduce a ribozyme that specifically cuts and degrades that mRNA.
That's the idea. It's a form of gene therapy at the RNA level. Selectively silence the expression of harmful genes by destroying their messengers. It's a very active area of research with huge therapeutic potential.
Okay, so we've seen how vital nucleotides and nucleic acids are. It seems logical then that messing with them chemically could be a powerful way to treat diseases.
Absolutely. By creating synthetic molecules that look like natural purines, pyrimidines, nucleosides, or nucleotides but have some chemical modification, we can often trick cellular enzymes.
How does that help?
Well, these analogs can get incorporated into pathways that use the natural molecules, but because the analog is slightly different, maybe it's missing a key group or has an extra one, it can block the pathway. It might inhibit a crucial enzyme or get incorporated into growing DNA or RNA chains and stop them from elongating further.
So they act as inhibitors or chain terminators.
Exactly. And this strategy has led to some incredibly important drugs.
Let's look at some examples. Gout. You mentioned allopurinol.
Right. Gout is caused by excess uric acid, which is the end product of purine metabolism. Allopurinol is a synthetic analog of hypoxanthine, one of the purine bases.
How does it work?
It inhibits a key enzyme called xanthine oxidase, which is responsible for converting hypoxanthine and xanthine into uric acid. By blocking this enzyme, allopurinol reduces uric acid production, relieving the symptoms of gout.
Makes sense. What about cancer therapy? That seems like a prime target for blocking DNA synthesis.
Definitely. Many chemotherapy drugs are nucleotide or base analogs. They target the fact that cancer cells are usually dividing much more rapidly than normal cells, so they're constantly synthesizing new DNA.
Examples.
A classic is 5-fluorouracil or 5-FU. It's an analog of uracil and thymine. Inside the cell, it gets converted into a molecule that irreversibly binds to and inhibits nimetylate synthase.
The enzyme that makes thymine nucleotides for DNA.
Precisely. If you block the metylate synthase, the cell runs out of DTMP, thymine deoxymonophosphate, one of the essential building blocks for DNA. DNA synthesis halts, and the rapidly dividing cancer cells die.
Clever and brutal.
Any others? 6-mercaptopurine, 6-MP, and its relative azathioprine are curine analogs used both in cancer therapy, especially leukemias, and as immunosuppressants.
Immunosuppression, how does that work?
Immune responses, like rejecting a transplanted organ, involve the rapid proliferation of immune cells, lymphocytes. Azathioprine gets converted to 6-MP, which then gets incorporated into nucleic acids or inhibits purine synthesis pathways, ultimately slowing down the proliferation of these immune cells and suppressing the rejection process.
Okay, and finally, the really big breakthrough area for analogs was antiviral therapy, especially for HIV/AIDS. These often targeted the sugar part, right?
Yes, modifying the sugar turned out to be incredibly effective against viruses that use reverse transcriptase, like HIV. The most famous example is zidovudine, better known as AZT.
What is AZT an analog of?
It's an analog of deoxythymidine, the nucleoside containing thymine and deoxyribose.
And the modification?
The crucial change is at the 3' position of the deoxyribose sugar. Normally, there's a hydroxyl H group there, which is absolutely essential for forming the next phosphodiester bond to elongate the DNA chain. In AZT, that 3OH group is replaced by an azeto group, NASN3.
So what happens when the virus tries to use AZT during replication?
HIV uses an enzyme called reverse transcriptase to copy its RNA genome into DNA. This enzyme readily picks up the activated triphosphate form of AZT and incorporates it into the growing viral DNA chain, thinking it's a normal thymidine nucleotide. Then, because there's no 3OH group, the next nucleotide cannot be added. The DNA chain cannot be elongated further. AZT acts as a chain terminator.
Stopping viral replication cold.
Exactly. And drugs like AZT and didanosine, DDI, another chain terminator analog, were revolutionary because the viral reverse transcriptase is much more likely to incorporate these faulty analogs than our own cellular DNA polymerases are. This gives the drugs a degree of selectivity against the virus. It was a landmark achievement based on understanding nucleotide chemistry. Hashtag tag outro.
Wow, we've covered a huge amount of ground. From the tiny chemical details like tautomerism ensuring the right base pairs and the charged phosphates.
All the way up to the elegance of the double helix, Chargaff's rules making sense of it, and that unbelievable packing problem, 2 meters of DNA squished into a tiny nucleus via histones and nucleosomes.
And then exploring RNA, the messenger, the transporter, the structural core of the ribosome, and even an enzyme itself.
Right, the ribozymes, plus the variations like zDNA, the clinical relevance of G tetraplexes, and how we exploit nucleotide chemistry to make drugs like AZT.
Yeah.
It really shows how deeply chemistry underlies all biology.
It really does. And thinking about those ribozymes, that potential ancient RNA world, leaves us with a really fascinating final thought for you, our listeners.
If RNA really could function as both information storage and catalyst, essentially the basis of early life, before proteins even evolved, what does that truly suggest about the fundamental capabilities of this molecule?
And if we're now learning to engineer ribozymes to target and destroy disease-causing RNAs, what other biological frontiers might open up if we could harness that ancient catalytic power of RNA in new ways? Could we repair, rebuild, or redesign biological systems at an even more fundamental level?
Something profound to think about. What are the ultimate limits and possibilities hidden within the chemistry of RNA?